All right, class. Okay. So we have learned that if we change the strength of the stimulus, the size of action potential does not change, right? So what changes? If a stimulus is a very, very strong one, it will cause the action potential to occur more frequently. So the strength of the stimulus changes the frequency of the action potential. So if you have, and there is a direct correlation between these two. If you have a stronger stimulus, that will lead to, if you have a strong stimulus, you will have greater frequency of action potentials. Now, let's play a game. Gently touch your skin. You don't feel any pain, right? But there are action potentials being generated, and that's why you can feel that you are touching your body. Now, press a little harder. You don't feel that pain, right? Still, you can feel the pressure because right now, with a stronger stimulus, probably the frequency of action potentials are more. Now, pinch yourself. Pinch yourself hard. When you pinch yourself hard enough, that's a super strong stimulus. And that causes a lot of action potentials like boom, 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 boom. And so many action potentials will help your central nervous system to perceive it as pain. Does that make sense? All right. So let's look at the diagram. We do see the strength of stimuli at the bottom as shown by the red step-like structures, and on top we do see the frequency of action potentials as shown by the black lines. Now, when the stimulus is not super strong enough, as shown by the small step here, we don't see an action potential. Increasing the strength of the stimulus, we do see some action potentials here. And when you increase the stimulus, or when you increase the strength of the stimulus even further, this is like pinching. Obviously, you have so many action potentials bombarding your central nervous system. Boom, 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 and you feel it. Okay? So we understand that by increasing the strength of the stimulus, the size of action potential does not change. However, the frequency of action potentials depend on the strength of the stimulus. All right. Now, let's talk about refractory periods. Now, we understand that action potentials can only increase in frequency up to positive 30. It cannot go beyond positive 30, right? And there is a refractory period after an action potential where neurons cannot become excited again. So there are two types of refractory periods. Now let's look at the tan colored area. The tan on the diagram, let's look at the tan colored area. During this time is called absolute refractory period. What do we mean by that? During this time, the action potential is in progress, right? So during this time, we have action potential in progress. And when the action potential is in progress, know that there cannot be another action potential. So absolutely not. Can there be another action potential during this time? Absolutely not. Okay. So that's absolute refractory period, which absolutely does not allow a second action potential to be generated in this time frame. And why is that? For the action potential to take place, the voltage gated sodium channels need to open up, right? At threshold, you have to have the voltage gated sodium channels open in order for depolarization to take place. However, during the absolute refractory period, specifically at this point when the cell is repolarizing back to resting membrane potential, at this time, how are the voltage gated sodium channels? Remember, they are inactive. Inactive voltage gated sodium channels. And listen carefully. When the voltage gated sodium channels are in inactive state, no matter how strong another stimulus is, doesn't matter how strong another stimulus is, these channels cannot be opened. Okay? Okay, so if the voltage gated sodium channels are closed, can a strong stimulus open it? Open it. Yes. If the voltage gated sodium channels are inactivated, can a strong stimulus open it? Can a strong stimulus open it? The answer is no. Therefore, during absolute refractory period, you cannot have another action potential. Absolutely not. Okay. Now, there is another refractory period that is called the relative refractory period. And let's talk about it now. The relative refractory period is when the potassium channels are still open and the voltage gated sodium channels are kind of resetting them back to closed state. So during this time, the potassium leak channels are open and some of the voltage gated potassium channels are also open, and they are kind of resetting themselves back to closed state. Okay, and in this period, and also some of the voltage gated sodium channels, they are resetting them back to closed state. Okay, so kind of some closed, some open voltage gated sodium channels. And at this stage, when you have a strong stimulus can actually generate another action potential during relative refractory period. So during relative refractory period, you can have another action potential, provided you have a strong stimulus. Okay, all right. Let's talk about conduction in an unmyelinated neuron versus conduction in a myelinated neuron. Now, one thing that you must know that conduction rate is much slower in an unmyelinated neuron as compared to a myelinated neuron. Action potentials are produced down the entire length of an axon. So let's draw this. So we have action potentials generated everywhere. Okay. And the conduction rate is slow because so many action potentials are generated and each one is an individual event. Okay. However, if you look at a myelinated neuron, which is right here, the myelination provides insulation. So the idea is the action potential that is generated at one point cannot dissipate. And that increases the strength and speed of the action potential. The only point where sodium can leak out and potassium can leak out. So sodium can come in, potassium can go out. The only points are the node of RAN-VA, which are right here, right? At the node of RAN-VA, only we have in and out movement of sodium potassium. So sodium ion channels are very, very concentrated at these nodes, and as a result of these, the action potential kind of hops from one node of RAN-VA to another, as shown here. As you see, the action potential jumps from node to node, and that's what makes it a faster process, and this is called saltatory conduction. So know that in case of myelinated neurons, the action potential jumps from node of RAN-VA to another node of RAN-VA, and this is called saltatory conduction. And it is only at the node of RAN-VA that movement of ions is allowed, and the sodium channels are concentrated at the node of RAN-VA. All right. So this is just a summary slide. It does say that if you increase the diameter of the neuron, that reduces resistance. Therefore, you will have faster conduction of action potential in if the axon is thicker. So let's draw. Versus this one. So in this case, you have a larger diameter, and that reduces resistance. Remember, resistance, conductance, we talked about these terms. So if you have less resistance, the rate of flow will be faster. So faster conductance. However, look here, the diameter is narrow, so you have slower conduction. And also it tells us that if an axon is myelinated, it's faster in case of a myelinated axon. In case of a non-myelinated or unmyelinated axon, the rate of conduction is much slower. All right, I'll start a new video with synapse.